So now it's time to talk about cell boundaries because they're so important to the chemistry of cells. The cell membrane is actually a really delicate layer. It really doesn't have any strength, but it has a really important function. It regulates what gets in and out of cells. It is a barrier, and it doesn't just passively determine what goes in and out like just a door, but it actually forms a barrier and it can move substances in and out of the cell because it can have pumps in it. So the backbone of the cell membrane is a phospholipid bilayer, and there are proteins that move around in it. Some of them don't move because they're stuck to the cytoskeleton. Other ones can move around. This model of the cell membrane we call the fluid mosaic model, which was first proposed by Singer and Nicholson in 1972. And their model describes a cell membrane as a mosaic of proteins scattered on a phospholipid background. Now, phospholipids are weird in that they're polar, and they have both a phosphate end and a nonpolar hydrocarbon end. Now, the phosphate end is polar because it's got phosphate and it's charged. The hydrocarbon end is just a string of carbon atoms with hydrogen around it, and that's totally nonpolar. And as it turns out, the fatty acid tails of the bilayer, it's two layers of phospholipids, face each other because they're nonpolar and they love each other. And the outside of the cell membrane, both facing into the cell and outside of the cell, is polar phosphates. So if I go down to this diagram down below here, each phospholipid, as you see in this picture, is an orange ball and two little tails attached to it. This is one phospholipid right here. This is the little circular ball. This part right here is the hydrophilic phosphate. Hydrophilic means water-loving. This is that part right there, or this part right here. And these two little arms facing down are these two little yellow threads dangling down. As it turns out, a cell membrane is two layers of these phospholipids. The little dangling phosphate or fatty acid tails face each other because they're nonpolar and they hate water. Water is out here, water is in here. And things that are nonpolar don't like water because they can't hydrogen bond with it. So the fatty acid tails face each other. These little green ball strings, those are proteins that are wedged in the cell membrane. They're the mosaic. And most of them can move around. This is another protein. It's a surface protein. There's another protein there. If I'm not mistaken, actually, these little green balls, those may be sugar saccharide chains. The artist doesn't indicate, but there are attached to a number of the proteins, there are carbohydrate chains for markers present. And so, yeah, they call these, this is a glycolipid. So these are glycolipids, meaning it's a carbohydrate with a little fatty acid tail here. And so the fatty acid tail hangs down in the core of the membrane, which is nonpolar, but the carbohydrate part's polar and it sticks out like deer antlers. This is a protein, a protein, a protein. So on the top layer up here, the orange layer or red, those are the phosphate heads. Down here, this reddish layer, phosphate heads. In here, this bright yellow zone, those are the two layers of the fatty acid tails that face each other. Everything that's in this region is nonpolar. So this protein is stuck. Part of it is stuck in this region. So this region of the protein is nonpolar, and that's why it's there. The surface part's polar here. The surface part's polar there. The part in the middle is nonpolar. That's why it's there. This particular protein is a channel protein. You'll notice it's like a tube. It allows chemicals to shoot through the cell membrane that otherwise could not go through. This protein has a long coil sticking out. These tend to be marker proteins that indicate what the cell is. So cell membranes are actually kind of complicated. In addition to all that, most cell membranes have cholesterol. This little yellow structure right there is cholesterol. Most of your cholesterol lives in your cell membranes in the core. This is called the core of the membrane. So this bright yellow region through here is called the core of the membrane. It's very nonpolar. And it holds all the nonpolar stuff that you eat or are exposed to. The fatty acid tails are in that core of the membrane, and that's why it's nonpolar. So that's the structure of the cell membrane. It's complicated. It's actually much more complicated than the cell wall. All the cell wall is rods of cellulose that are all tangled and piled on top of each other. What we have here is a cell membrane down here, which is this stuff here, right here. This is down here in this blue layer. But then these rods that are laying on top, those are just rods of cellulose that surround a plant cell and reinforce it. It's just like wrapping screen around something to protect it. And that's really all a cell wall is. Cell walls don't do gatekeeping. They don't do pumping. They just wrap around things and keep them safe, more or less. Cell walls are made of cellulose, and that's what these rods are. They're just rods of cellulose. Now, we have to talk about diffusion and osmosis. So it turns out that chemicals tend to move from where they are concentrated to where they are not concentrated, and they tend to just spread out. So if you dump a dye into a beaker of water, it's all concentrated in one spot at first, but it tends to all spread out. And the reason for that is the water molecules are bouncing around randomly, and they hit the dye molecules. The dye molecules are bouncing around randomly, and they all spread out. We call that diffusion. So if you wanted to measure concentration, that you did that last year, that's the mass of a solute in a given volume of solution. So for example, sugar in the human bloodstream is measured as milligrams per deciliter. So, and that's an important thing to determine whether or not you have diabetes. So whenever you get a physical and blood is taken, they measure your sugar in milligrams per deciliter to make sure that your blood sugar is in the right level. Now, diffusion is simply then the movement of things that are highly concentrated in one place to an area where they're less concentrated. So diffusion is technically the movement of substances from high concentration to low concentration caused by the random motion or the random kinetic energy of particles. So if you'll notice in this diagram to the right here, we have a bunch of particles out here, and here's a cell membrane. Each one of these little green balls is a phosphate head, and the two little tails hanging off of each green ball are the fatty acid tails. This is the core, and these are the phosphate heads. These little balls will move through the membrane, and they will evenly spread themselves out. So over time, you'll end up with an equal concentration of the chemicals on both sides of the membrane. This requires no energy on the part of the cell. This is how oxygen moves into your red blood cells. When you take a breath of air into your lungs, oxygen is like this. Your cells are like this in your lungs. So the oxygen moves from the air into your blood cells in about 1 second. It goes to this, what you see right here. The air that you breathe out has oxygen in it, but the blood also has oxygen in it because the oxygen diffused there. Now, when things are equal, we call that equilibrium. Okay? But at equilibrium, that doesn't mean things are sitting still. It means this particle shoots in here, but then this particle shoots out there. Things move in and out at the same rate when things are at equilibrium. Now, most membranes are permeable to something. That means they're selectively permeable, meaning they let some things through but not others. And cell membranes are no exception. Some membranes are selectively permeable membranes. They let some things go through, but then they don't let other things go through. Water is one of the most important commodities that has to get through cells. And there's a special name for the movement of water through a selectively permeable membrane, and that's called osmosis. Water passes really easily through membranes because there are special proteins in membranes called aquaporins that let water go through really fast. Now, cells can find themselves in problems if water goes through too fast. If too much water exits a cell, the cell dehydrates and dies. If too much water enters a cell, the cell will swell and potentially burst. So if cells are bathed in pure water, the more water is going to... So if you put red blood cells in a pure water solution, let's see if we've got a situation here. Yeah, let's go over here. This is a plant cell that's in pure water. When you have pure water around a cell, more water will try to enter the cell than exit the cell, and the cell will swell. So you can see the plant cell here, the black part, that's the cell wall. With cellulose, it's swelling outward under pressure. Okay. Now, if you instead put the cell in salt water, the opposite happens. If there's salt water out here, the salt interferes with the water moving into the plant cell, but water will still move out. So as water moves out of the plant cell, the whole cell shrivels up. The wall won't change shape, but the membrane will. And what you see here is a tiny little vacuole, and the cell is all shriveled up into a tiny space. This is the way a plant cell is supposed to exist. The plant cell is supposed to be bathed in not pure water, but like rainwater. Rainwater has some solutes in it, which is healthy. So that means that there will always be water moving into the plant cell and water moving out of the plant cell. And we call that water, with respect to the plant cell, isotonic. If you put a plant cell in a hypertonic water solution, more water will exit than enter, and the cell will shrivel. If you put a plant cell in a hypotonic solution, meaning it's pure water, more water will enter the cell than exit, and the plant cell will swell to the point of almost rupturing. And you'll notice the vacuole here is huge because that stores most of the water. Hypertonic means lots of chemicals. Hypertonic, if you put a plant cell in a hypertonic solution, you're putting it in a salty solution. Hypertonic is not referring to water, it's referring to salts in the water. Isotonic means equal salt levels. So if you put a plant cell in an isotonic solution, water will move in and out at the same rate and the cell doesn't change shape. If you put a plant cell in a hypotonic water solution, more water will enter than exit because there's absolutely no salt outside the cell, but there always is a little bit of salt inside the cell, and as a result, the cell will swell. If you put the plant cell in a salty hypertonic solution, the cell shrinks because more water exits than enters. The same osmotic situations are true for red blood cells. Put red blood cells in a hypertonic solution, more water exits than enters, cell shrinks. If you put a red blood cell in an isotonic solution, equal amounts of water exit and enter, the cell doesn't change shape. If you put red blood cells in pure water, the water rushes in and the cells rupture. In fact, if you inject pure water into somebody's veins, all of their blood cells pop and they die. So when you're at the hospital and they put a tube in your vein and they put fluid in, they're putting isotonic fluid into you, not hypertonic and not hypotonic fluid. So if you put a cell in a hypotonic solution, the cell can burst. We call that lysis. If you put a cell in a hypertonic solution, then the cell can shrink and undergo plasmolysis. That's what we call shrinkage of a cell due to water loss. That's called plasmolysis. So cells are kind of worried about what kind of water they're swimming in. And that's the reason why if you're eating a meal and you eat a salty meal, you get real thirsty afterwards. The reason for that is the salty fries are hypertonic. And when you put them in your digestive tract, you actually pull water out of your bloodstream into your digestive tract. That fires off the thirst receptors in your brain. You get thirsty, you drink water. Eating a salty meal always results in gaining a couple of pounds of weight because it's water weight. You have to drink water to counteract the hypertonic nature of salty food. You have to balance out the osmotic properties of the food and make it isotonic to you. And the only way to do that is to drink water with it. Now, water moving from one place to another in plants can create pressure. We call that osmotic pressure. So water moving in and out of cells creates pressure. If a cell wall is present around the cell, that can resist pressure, and that can prevent the lysis of a cell. But cell walls actually don't prevent plasmolysis. This is a typo. They prevent lysis. I'll have to change that. Animal cells have no such protection. So cell walls do prevent lysis, but our cells, animal cells, have no cell walls. So if you put any of your cells from inside your body into a pure water solution, they just pop. Your skin protects you. And by the way, you've probably noticed that if you get your feet wet for too long, like if you're walking in wet socks and wet shoes, the skin between your toes especially, it's really thin and delicate. It'll just rub right off between your fingers, and then you'll get blisters on your feet. Your skin will swell as it absorbs the water. It loses its strength, and then the skin will rub off. At that point, then you've lost your protection, and you can absorb water then through that open wound and microbes, and it'll often get infected. Now, so besides water, other things have to move in and out of cells. A lot of chemicals move in and out by facilitated diffusion. That's because cells have proteins in the membrane they create channels to allow chemicals to move in and out. There are channel proteins, and then there are carrier proteins. This is a carrier protein, and this is a channel protein. Channels are just tubes. Carriers are actually interesting. They're actually a cup. that'll accept a chemical from one side, and then the cup actually kind of flip-flops and shoots the chemical out to the other side. These are essentially doorways. This is like a revolving door, and this is just an open door. And so they don't expend any energy on the part of the cell, but all the cell can hope to do is get equilibrium here. The cell can never get more inside or outside because things are just randomly moving in and out. So if you're just creating a door, you're not forcing people to enter a room. They've got to want to enter the room. So we call this facilitated diffusion. It uses no energy. It doesn't achieve anything more than equilibrium, but it's how a lot of chemicals move in and out of your cells. Insulin serves as a facilitated diffusion protein to move glucose in and out of your cells, and that's why it's so important to you processing sugar energy. Cytochrome P450 is a famous protein on your red blood cells, which moves oxygen in and out of your red blood cells when the red blood cells are in your lungs, especially. So, facilitated diffusion is very common on cells. But that's not the only way that things move in and out of cells. There's also active transport. And the difference here is active transport is actually a pump. It's like a fan. Unlike a door doesn't actually force things to move, but a fan does. Active transport is an energy-requiring process that moves solutes from one place to another against their diffusion gradients. Instead of from high to low, it moves them from low to high. There's two basic ways to do this. The first way is what's being animated here. This is where chemicals, this is the active transport protein right here. It grabs chemicals, energy is used, it throws the chemicals through the membrane, and then it goes back to a resting state. So let me explain more about that in just a second, but let me go through the basics here. The sodium-potassium pump is the most famous active transport pump in the human body. It moves sodium out and potassium into nerve and other cells. It consumes more energy than any other pump in the human body. In fact, it's one of the most energy-requiring processes in the body, period. So, by pumping sodium out of a nerve cell, the outside of the nerve cell becomes positive. That sets the stage for the nerve cell to be able to send nerve impulses. And that's why the pump actually works, to set up nerve impulses. But the thing is, during a nerve impulse, the sodium that was pumped out passes back in through the cell membrane. Well, in order for a nerve to continually be able to pass nerve impulses, the pump has to pump the sodium out as soon as it leaks in. So, and to help the nerve cell maintain its positive charge outside, potassium gates allow potassium to leak out right after a nerve impulse. So the sodium facilitated diffusion proteins that let sodium go back into a nerve cell after the potassium pump pumps them out tend to work in a wave-like fashion. So let me go over this now. The yellow thing is the sodium potassium pump. This is a sodium gate, and this is a potassium gate. Now here's how this pump works. Let's start at the beginning of the cycle here. In fact, let's go. I don't have the animation handy here, so let's see if I can find it quickly. And you'll watch as I go to this. So I'm going to BioBook, I'm going to Special Topics, and let's go to Tutorials. And then let's go to the nervous system, and then let's look at the sodium-potassium pump here. So here it is. I'm going to step through it for you. Right now the pump is primed. Sodium is loaded here, potassium is loaded here. This is an ATP molecule being delivered from a mitochondria. The ATP molecule comes up to the sodium-potassium protein and drops off this last phosphate. That phosphate contains energy. That causes the pump to throw the sodium out and the potassium in. So you notice it throws them, shoots them out. Now, potassium is now inside that was outside. The sodium is now outside that was inside. The phosphate here is just going to be released. Watch, it'll just kick it back out. Now here's the funny part. Watch the sodium come back in. This is what a nerve impulse is. A nerve impulse is this right here is a sodium gate. When that sodium gate opens, that's a nerve impulse. The sodium leaking in is what causes the nerve cell to be able to conduct an impulse. But now all of our positive charges are gone from out here. So in an effort to restore the positive charge on the outside, the nerve cell leaks the potassium, which is positive, back out to the outside. Potassium leaks out. Now the outside is somewhat positive, and the nerve cell can get ready to fire another nerve impulse. But to keep things charged, ultimately, the pump has to rethrow the sodium out and the potassium in. And to do that, it has to use more ATP energy to throw the sodium out and the potassium in. So this pump tends to work as a cycle round and around the clock. So let's go back to the animation now. This is the GIF animation. The pump works all the time. This is why your brain consumes so much energy all the time. Sodium is being pumped out. Why? Because every time you think a thought, the sodium leaks back into your nerve cells. So in order to think the next thought, the sodium must be pumped out of your nerve cells. The potassium here is just going along for the ride to help recharge the outside positive nature of a neuron after the sodium leaks in. So that is the sodium-potassium pump. We'll talk more about it in class. It is the most important active transport pump you have. You'll notice all these things here, phospholipids. This is a phosphate head, and these are the two fatty acid tails. This is the surface of a neuron. This is the fluid mosaic model. These are phospholipids. The green, the yellow, and the purple things are proteins. They are the mosaics in the fluid mosaic model of the cell membrane. Now, the other way, there were two types of active transport, if you remember. This is the first type, where you have proteins that pump chemicals through the membrane. The other way is endo or exocytosis. This is where a whole cell is engulfed. This is like a white blood cell right here, and it's eating a microbe, and it digests the microbe with a lysosome. So let's watch it from the top here. Here comes the little microbe. It bumps into the white blood cell. The white blood cell surrounds it. It eats it, and then a lysosome fuses with this little food vacuole here that this bug is trapped in, and then it's digested. Now, in the story here, they call this an amoeba, and you can think of it as an amoeba, but this is exactly how your white blood cells work too. This is called phagocytosis. So let's go from the top one more time. Now, when the small ciliate, this is a ciliate, it's got cilia, when it first bumps into the amoeba or the white blood cell and it's engulfed, that's called endocytosis. That's absorbing a cell. And then ejecting the undigestible parts, that's exocytosis. So that's both endo and exocytosis right there. In general, though, biologists call this phagocytosis, the engulfment of one cell by another cell. One cell is eating another cell. Right here, these are white blood cells right here that are eating these rods. These are bacilli. So I think these are bubonic plague bacilli. These are diplobacilli. If you look in a sample of blood and you see these big long rods, you're going to be taking some antibiotics immediately because that's bubonic plague, and the patient's going to be rushed to ICU and be given special antibiotics. Anyway, that's the end of the discussion on membranes. The last part's just on the unity and diversity of life.